So you're going to look at autosomal linkage. Okay. Now, if you look at this diagram, you can see that in this chromosome, you've got gene 1. And then in the second chromosome, you've got gene 2, gene 3, gene 4. Now, you know that these two chromosomes are not homologous, isn't it? Because homologous chromosome must be chromosome having the same gene in the same locus. But these two chromosomes don't have the same gene, right? In the same locus. So they are not homologous chromosomes. And therefore, they shouldn't have the same length. Huh? Correct or not? They should not have the same length, okay? Right. So you can see that in this chromosome, you got gene 1, this chromosome, gene 2, gene 3, gene 4. So gene 1 is not located in the same chromosome as gene 2, gene 3, and gene 4. So gene 1 is an unlinked gene with gene 2. It's unlinked gene with gene 3, is an unlinked gene with gene 4. Whereas gene 2, gene 3, gene 4 are linked genes. Linked genes means those genes are located in the same chromosome. So they're linked within the same DNA, right? They're linked together in the same DNA. So that's the meaning of linked genes. Genes that are found in the same chromosome. So the set of genes at different loci, but they are found on the same chromosome, they are considered as linked genes. So G1 gene is not linked to G1. Gene 1 is not linked to gene 2. Okay, it's not linked to gene 3, it's not linked to gene 4. So it's called unlinked genes. Okay. So linkage means when you have two genes on the same chromosome, now they tend to be inherited together. You see, when gene 2, gene 3, gene 4 are located in the same chromosome, they tend to be inherited together. Okay, but if they are unlinked genes, they can be inherited separately. Okay, so we're going to look at an example here. What do you mean by inherited together and inherited separately? Okay, I'll draw and show you so that you understand better. Okay. So I'm going to show you two genes that are not linked together. Okay, so let's say gene A and gene B. So can you see here, I've drawn two genes. A, this is one type of gene, and B, this is another type of gene, right? So gene A and gene B are unlinked genes. They are found in different chromosomes. So because of that, this pair of homologous chromosomes will independently assort during metaphase. This pair of homologous chromosomes will independently assort during metaphase 1 also. You know, do you remember what is independent assortment? How this pair of chromosomes arranged at equator is independent of this pair. Okay, so that means to say that now when the chromosome arranged like this, big A is on the left, small a is on the right. But they can arrange in the other way around, right? Where small a is on the left, big A is on the right, isn't it? Same as this pair. It can be arranged big B on the left, small b on the right, or small b on the left, big B on the right. So because both pair of homologous chromosomes, they can arrange independent of the other pair, then after meiosis, you'll find that you'll get 4 different types of possible gametes, isn't it? Hello? You will get 4 different types of gametes, okay? You know how to calculate? This pair got 2 ways of arranging. This pair got 2 ways. So altogether, 2 times 2 is 4. Okay. 4. So you get 4 types of gametes. Okay. But imagine now that A and B, these 2 genes are linked, right? Linked. So big B is here, and small b is here. Okay, so if A and B are linked gene, then what are the possible different type of gametes that you can get? How many number? 2 or 4? 2 or 4? Please put your finger up. 2 or 4? Yeah, the upstairs. 2, isn't it? 2, right? Okay, thank you. This one go this side. Let's see. This one go this side. So one gamete take this type. Another gamete take this type. Isn't it only 2 types? Compared to if it's unlinked genes between the A and B, then you've got 4 types. So can you see the effect of linkage where the genes on the same chromosome will most probably be inherited together, and then because there's no independent assortment between these 2 genes during metaphase 1. Okay, so linked genes will be inherited together. So that will reduce the genetic variation in the gametes, right? For linked genes. Instead of 4, there are 2. Can you see very clearly? Okay, right. So that is introduction. Now is the real one. Okay, so let's do the genetic cross for linked genes. The way you write the symbol will be slightly different. Okay, so let's big E be the allele for striped, small e be the allele for ebony. Striped means, you see here at the abdomen of this Drosophila, which is actually fruit fly, it's got stripe on it. Ebony means totally black body, so you cannot see the stripe. So this ebony, totally black. Okay. Let big A be the allele for normal antenna. So this one, can you see the antenna here? The tiny little fiber structure, which is branched. So this is the antenna, is normal. Small a be the allele for Aristopedia antenna. Aristopedia means appendages. So what does it mean for Aristopedia antennas? You look at the antenna for this one, is like the foot growing on the head, forming the antenna. So this is called Aristopedia. Okay. So the gene for E, which is the pattern at the abdomen, and the gene for A, which is the pattern of the antenna, they are linked genes. So when you do the genetic crosses for linked genes, everything's the same except that you have to put the bracket to bracket the 2 linked genes together to show that they are found in the same chromosome. Okay? So therefore, striped normal cross with ebony Aristopedia. We start with offsprings that are pure breeding, means homozygous with contrasting characteristic. So big E and big A must be placed together in a bracket. Okay, small e and small a placed together in the bracket, showing that they're linked genes. So this is the only difference when you do genetic crosses for linked genes. And the rest will be the same. Okay, gametes. And then F1 genotype. Okay, so this is heterozygous. Genotype is heterozygous. Phenotype is striped body and normal antenna. So all the dominant, coded by the dominant allele. Okay. So continue with the cross. So continue with the cross, right? Take F1 to become P2 and do the cross. So all P2 are heterozygous dominant. So after you do the cross, you find that the phenotypic ratio is actually 3:1. 3 striped normal antenna and 1 ebony aristopedia. So only this is ebony aristopedia. These 3 are normal antenna and striped. Okay. So this one is showing you that this is normal antenna and this is aristopedia antenna, which looks like the foot, right? Growing to become the antenna. So that's why it's called aristo, stands for appendages. Okay, aristo stands for appendages. So do you see that this 3:1 ratio is like the normal Mendel's monohybrid ratio for F2 generation? Because even though you're looking at 2 characteristics coded by 2 pair of genes, but these 2 pair of genes are found in the same chromosome. So we are looking at inheritance of the genes from just 1 pair of chromosomes instead of 2 pairs. That's why the phenotypic ratio in the F2 is only having 2 different phenotypes instead of 4. So let's do some test cross. Now, don't fill in the blank yet. We have to do the test cross in order to fill in the blank. Okay? Remember what is test cross? Do you remember what is test cross? Genotype. When you see the phenotype of an individual is showing characteristic coded by dominant allele, for example, brown eye. But you know that in our population, brown eye can be big B, or big B, small b, right? So you want to know whether this individual with brown eye is homozygous or heterozygous. So you do test cross. Okay. So let's say if you are doing test cross on these characteristics. Okay. Now, for 3 cases, the offspring phenotypic ratio will be different. Now, if the 2 genes are not linked, so you do the test cross on a dihybrid crosses, when the 2 genes are not linked, what will you get in the offspring phenotypic ratio? If the 2 genes are linked, then what is the offspring phenotypic ratio? And the case number C is when 2 genes are linked and crossing over occur, what will be the offspring phenotypic ratio? Okay, so you write it down first. After that, you do the working to find out. Okay, write down the ratio first. Okay, so let's do the cross together. So first case scenario is just a normal dihybrid cross. So you just do the cross as usual because the gene for E and A are unlinked genes. So just do the cross as usual. Okay, so once you do the test cross, you can see that I'm using a heterozygous dominant to cross with a homozygous recessive as a test cross. So you'll find that the offspring phenotypic ratio is actually 1:1:1:1. Okay, can you see that? This is striped and normal. This is striped and aristopedia. This is ebony and normal. This ebony and aristopedia. So altogether, we've got 4 different phenotypes, each 1:1:1:1 ratio. Okay, so this is the offspring phenotypic ratio for dihybrid crosses for unlinked genes, where this individual is heterozygous dominant. Now we look at, okay, so same cross, but these 2 genes are linked genes. What will be the outcome? Okay, the rest are the same. The only difference is that they are the linked genes, the E and A. So how do you do your cross now? Right? The way you write your symbol is different, right? Okay, so because E and A are linked genes, so you have to put bracket to link them together. Okay, so once you do the cross, you look at the outcome. So the outcome is, you can see that these two are striped and normal. These two are ebony and Aristopedia. So that means the offspring phenotypic ratio in a test cross between heterozygous dominant and homozygous recessive in linked genes will be 1:1 ratio. Can you see that it's similar to monohybrid crosses already? Because even though you got two genes, these two genes are found in the same chromosome. So we are considering the inheritance of a pair of chromosomes only instead of two pairs in dihybrid crosses. So the outcome is similar to monohybrid cross because it's inheritance from a pair of chromosomes. Okay, dihybrid is two pairs, isn't it? Just now I draw two pairs. Now this one is only one pair. Okay, so moving on further, which is the last case study, is we know that homologous chromosomes can cross over, right? Homologous chromosomes can cross over. So this can happen for linked genes. Okay, now I've drawn two cases here, left and right, right? Both are linked genes. So when there's no crossing over, after meiosis, you will get each of the gamete taking either this chromosome or this chromosome. And then the sister chromatids are also separate, okay? But when there's crossing over, there'll be 4 possible types of gametes already. Because you've got the original parental type of allele combination, which is big E, big A. You will also have got recombinant type of allele, which is big E, big A, small e, big A. So the big E, big A, and small e, small a are the recombinant type. Recombinant means recombine. That means the two alleles come from two different sources, they combine together. They rearrange and combine together. So these two are recombinant type. These two are the parental type, the original one. So these two types of genotypes in the gametes arises due to crossing over. Okay? And crossing over occur by chance. So because of that, majority of the gametes will be big E, big A, small e, small a. Minority means small number of gametes will have got big E, small e, big A, because crossing over is by chance. So among all the germ cells that carry out meiosis, most of them there's no meiosis, but maybe a few of the germ cells that are meiosis, they are crossing over. Most of them no crossing over. So there will be large number of non-crossing over outcome, small number of crossing over outcome. Okay? Can follow? Right? So in this case here is there's no crossing over. All of them will be small e, small a. In this case here. This is for this one. Huh? I think crossing over also gives the same result, right? This one, if crossing over gives the same result, isn't it? Okay, so crossing, no crossing is the same. Okay, so therefore, if crossing over occur, we will get these 4 types of gametes. Large number are parental gametes, small number of recombinant gametes here. So these 2 are parental gametes from here. These 2 are recombinant gametes from here. So then you do the cross. So usually when you draw Punnett square, right, the gametes that you write on top here must belong to the first parent, and the gamete that you write on the side here must belong to the second parent. Okay, or this is for male gamete, this is for female gamete. Okay, so do the Punnett square, and then now you see that offspring phenotypic ratio. Phenotype first, okay, all these 4 phenotypes you know how to do. Then the parental classes will have got a higher number compared to recombinant classes will have a smaller number. Now this number you don't have to don't have to memorize because I just simply put a number to show that this is larger, this is smaller only. I just simply put a number to show you. Okay? But it makes sense, right? Recombinant number will be smaller because it's all based on crossing over, which is by chance. Chances of crossing over is smaller. So the number must be smaller. Okay? So large numbers of gametes will receive chromosomes having the original parental combination, which is big E, big A, or small e, small a. So they are the parental gametes. So there's a complete linkage. That means the big E and big A, they are linked together in the same gene, and they are passed down to offspring in the same, not same gene, same chromosome. Big E and big A are found in the same chromosome and passed down together. So this is called complete linkage. They do not separate. Okay. No separation of linked genes because no crossing over occurred to swap the genes between non-sister chromatids of homologous chromosomes. Okay. But small number of gametes will receive chromosomes having allelic recombination, big E with small e with big A. So these are called the recombinant gametes. And this is called incomplete linkage. Means the original linked genes, they are separated due to crossing over. So there's a breakage and separation of the original linked genes due to crossing over that swaps the genes between non-sister chromatids of homologous chromosomes. So there is an incomplete linkage in the gametes and therefore in the offspring. That means the original linked genes are not inherited together already, right? They break off already. Okay, incomplete linkage occurs. Okay, so therefore we have worked out the test cross outcome of the test cross between heterozygous dominant and homozygous recessive. For unlinked genes, the ratio is 1:1:1:1 versus linked gene, the ratio is 1:1. But if there's crossing over, then the ratio will be 2 of them larger number, 2 of them smaller number. And this smaller number represent the incomplete linkage. Incomplete linkage means there's crossing over. Okay, so that's all. I'll skip this because this is not in your syllabus. I'll skip this. You can read for your own knowledge. So what you have learned today is you have used genetic diagrams and Punnett squares to explain and predict the results of dihybrid crosses that involve autosomal linkage and epistasis, which is gene interactions. Okay. So that's all for today's lesson.